Good. And it's my great pleasure and honor to introduce our speaker today, Dr. Professor Liisa Galea. Liisa is the Treliving Chair in Women's Mental Health at the Center for Addiction and Mental Health in Toronto, after serving as a professor at the Javad Movafagian Center for Brain Health at the University of British Columbia for 25 years. She leads the Women's Health Research Cluster with more than 750 members worldwide. And Liisa is a world-renowned expert in sex hormones influences on brain and behavior in both health and disease states, with a focus on stress-related psychiatric disorders and dementia. Liisa's research goal is to improve brain health for all by examining the influence of sex and sex hormones in health and disease. She has won numerous awards. She's active in advisory boards, editorial boards, peer review panels internationally and nationally. And she's a tireless advocate for women's health research and for sex and gender-based analysis to improve mental health for all. On her homepage, she also is listing some fun stuff and facts, and one of them is that she's very proud of her Estonian Maltese heritage. And that actually, for me as an Estonian, was one of the not most important, but very important facts
I agree with you. It's the most important, probably.
to be a very persistent and bombing her with the emails to get her as Estonian, but also internationally renowned expert with us today here. So I'm very glad. And now over to you. Screen is yours, Liisa, please.
Thank you, CITE. I'm thrilled to be able to speak to you all. Every time I come to your part of the world, I start to tear up a little bit because I lost my mom about 10 years ago now. And my house, I grew up with Estonian grandparents and Estonian in the house. And I actually was quite fluent in Estonian, but I've lost most of it, right? If you don't hear it, you lose quite a bit of it. So I look forward to coming back someday and regaining some of those memories back again. So I've made this more of a sort of general discussion about a little bit about my journey, but about why I think sex and sex hormones matter so much for brain health. And let's see how much of it I get to go through. But before, I just wanted to acknowledge that I live in Canada, and most of the work that I'll be talking about was done in Vancouver, Canada, which is part of the unceded traditional and ancestral territories of the Coast Salish peoples, the Musqueam, Squamish, and Tsleil-Waututh First Nations. Today I'm in Toronto, Ontario, Canada, which is covered by Toronto Purchase Number 13 of 1805 with the Mississaugas of the Credit. And Toronto is now home to a vast diversity of First Nations, Inuit Métis, who enrich the city, and CAMH, which is the institution I'm from, is committed to reconciliation. I think it's probably wise for me to define sex and gender. I was at a conference where we finally realized that sex and gender don't always have different definitions or different delineations in different languages. And gender actually, even in English, is a relatively new concept. It was only created about 40 years ago. So most of my world is centered in terms of biomedical differences. So I'm talking about physiological characteristics that may differ between males, females, and intersex individuals. But gender is another psychosocial construct. Some people think about it as sexual orientation. It's not. It's whether or not you identify as a woman, non-binary, gender diverse, or man. And it's not even just about that. It's how society treats you and has expectations of you based on your gender identity. So here's my husband who identifies as a man, and this is how I expect him to act at the household. So it's not just at your work or school, it's also in your house. Um, I want to make it really clear. I'll be talking about disparities between males and females, men and women, but I want to be very clear that the disparities are much larger for people of color, Indigenous, trans, and gender diverse individuals. Each of those communities deserves attention and research devoted in their area. And I put some people who do some fantastic work in those areas. I'm sharing pictures from my own family, including my Estonian mother there. And I think I put this up to make me smile, but also to illustrate that there are probably a number of sex and even gender traits that you might notice right away. But you're probably very well aware that women on average tend to live longer than men, but you may not be aware that they're also more likely to live with chronic pain and disability. And my mom was no exception to that. She suffered from arthritis and a form of Parkinson's disease towards the end of her life. I really liked this study that came out using the Danish registry that showed for over 1,300 different disorders, on average, women were diagnosed 2 years later than men. And then we actually looked at how many of those disorders had a delay in diagnosis for women. And that was 996, so over 70% of the disorders. And if you add those all up, it's almost 4 years. Now, there are going to be many reasons for this disparity and the delay in diagnosis. Both on the biological side and on the gendered side of things. Often women are dismissed at doctors' offices, for example. But I think one of the major reasons is that most of our medical knowledge, including the way we diagnose people, is based on males or the experiences of men. So much so that symptoms in females for a variety of disorders are called atypical. And you see this atypical label across a wide variety of disorders that there might be a bias in the prevalence favoring males or favoring females, or no difference, like bipolar disorder in terms of who's getting the disorder. But I think it's really important to think about this because knowing that there's an atypical depression label for women and they're twice more likely to have the disorder, how is that atypical? It's obviously clearly not atypical. More of them are getting the disorder. And so to me, this suggests that there's a systemic bias. As I said, of course, gender is going to play a role. There's numerous studies showing that women struggle to get the right diagnosis. They're misdiagnosed. There's this delay in diagnosis, and they often are treated less seriously in the doctor's office. So basically, my take-home message is that males cannot serve as a default to females. To me, it's like we're using the wrong playbook. To me, it's like, we're trying to fix a refrigerator with an oven manual. Some things are going to be exactly the same, but some things are going to be completely opposite. And there are some suggestions that there are opposing molecular signatures between males and females for different disorders. As a neuroscientist, I, of course, care about whether or not there might be some differences in the brain. And there are. This is from a meta-analysis looking at gray matter volume. One thing I want to point out is that it's not that males always have larger brain volumes or brain nucleus volumes than females. You also see just as many areas that show the reverse. There are also some putative differences in white matter. There's more intrahemispheric connections in females and more intrahemispheric connections in males. Now, these may or may not contribute to the fact that we see sex differences in prevalence of brain disorders, and I put some very common ones out there, obviously the ones in males and men in blue, and the ones in women and girls in pink. And one of the things I really want to point out is that, and you might not have noticed it already, is that there's an age factor in there as well. In general, more of the male sort of dominant disorders occur earlier in life, and those in females occur a little later in life. And so that lifespan approach is also needed. Now, to me, this is interesting, but what's even more interesting is that we see sex differences in the manifestation of many of these conditions, even in conditions where we don't see a sex difference in prevalence, like schizophrenia or Parkinson's disease. There's a bit of a difference, but not as dramatic as what we see in Alzheimer's and depression. And I'll be giving you a couple of examples of this. So hopefully it's starting to become clear why I think it's so important to examine sex differences and influences and disorders. It gives us clues on how the disorder develops, the manifestation of that disorder, and the treatment. And that treatment is hardly ever studied. It also allows us to build better models of disorders, and that's true both clinically as well as preclinically. And better models are just going to give us better precision therapeutics. And obviously, if that doesn't convince you, our funding agencies are now mandating incorporation. I'm getting very worried about that, and maybe we can talk about that in the comments. I don't know how long that's going to last in the US. And but I know that Horizon Europe and Canada still follows these mandates. And I can tell you about, I actually haven't been a fan of the mandates anyway, and I can tell you about why that might be at the end as well if you have questions about it. If you do happen to see a sex difference in any data that you're looking at, it has implications for sex chromosomes. That might be what's causing it, sex hormones, or some combination of the two. And I thought I'd just give you this particular graph, even though I won't be talking very much about genetic differences, but the genetic similarities between two males, and I know I should have made this Game of Thrones, but this is from Ted Lasso. Hopefully some of you have seen Ted Lasso. The genetic similarities between, or differences between two human males is very similar, right? Is 99.9% similar. But what a difference that 0.1% can make in terms of phenotype. So knowing how large a difference that 0.1% can make in terms of phenotype, imagine now 15 times greater genetic difference. So between a human female and a human male, it's 1.5% differences in terms of genetic similarity. In terms of animal models, there are some 4 core genotypes, both in mice and rats. And so if you're interested in sex chromosomes and how they might be contributing to the trait that you're interested in, there's some really great models out there that you can use. Sex hormones also influence disease, and just so we're all on the same page, I'm showing you a picture of my adult children, and what I'm talking about here are estrogens, ovarian hormones like estrogens in females, and androgens such as testosterone in males. Obviously, we have each other's hormones. It's not that we, you only have one type of hormone. You don't, just in different concentrations. And these hormones act on hormone receptors that are located throughout the body, throughout the brain, not just in the reproductive tract. And obviously, it's much more complicated than that. Testosterone gets converted to the most powerful of all the estrogens. It's called estradiol, or a very potent androgen called dihydrotestosterone. And sex hormones themselves can also affect disease risk, symptomology, as well as treatment. And again, are hardly ever studied. I'm giving you an example from the schizophrenia literature showing across the menstrual cycle as estradiol levels decline, psychotic symptoms are increased. I think it's really important for me to talk about what studying sex differences is not. It's not sexist. It's not more complicated in one sex versus the other. It's not believing that males and females are sexually dimorphic or polar opposites, and it's not the last step. So what do I mean by that? All of us, it's really important for all of us to acknowledge that we have biases. And one of them is that, or notions. And so one of the things that I see in the literature is that a sex difference means that it's non-overlapping or dimorphic or opposite. That's not the case. There are many kinds of sex differences, and I'll show you that in just a second. Another one is we shouldn't study sex differences because that means that any sex difference we see in the brain means that female brains are inferior. I always think that's funny. Why is it the female brains are inferior? Why can't it be the male brains are inferior? Why does it mean, so just because there might be a gray matter volume difference, and even if it is lower in females and males, that doesn't mean that is inferior. That just means it's different. And in fact, there are many examples of where a decrease in gray matter is actually related to better behavior. And again, I can talk about that at the end if you'd like. The third is a notion I've already talked to you about, that females are somehow atypical. They're not atypical. And the last is that females are more variable because of our hormones. That's why they haven't been studied, or that's one reason. And there's been some really nice opinion pieces written about this. There have been meta-analyses in mice and rats showing that there's absolutely, across a large number of physiological, biological traits, there's no sex difference in variability. Now, I've seen this then explained to say, so sex hormones are not involved, or estrous cycle doesn't matter. And that's not what that means. That just means that the variability is the same between males and females. That what causes that variability within a sex might very well be due to hormones. And even in humans, this comes from some researchers in the Netherlands. They looked at brain structure volumes, and they found greater variability in males than in females. So it's not inherently that females are more variable. I really like to point out this slide, and that is that hormonal fluctuations are not a female problem. This is human males. This is testosterone. And this is showing you that there's a daily or diurnal fluctuation in testosterone levels. particularly in human males, and they decrease by as much as 50% every day. So given that there's a daily fluctuation in hormones in males and a monthly fluctuation in females, I have one question for you. Who's more hormonal now? Right? You can't say that one sex is more hormonal than the other. I alluded to this earlier, that there are a number of types of sex differences. I'm not going to talk about any of the three above there. I want to talk about what I'm calling sort of hidden sex differences. So what do I mean by that? There might be, and I see this a lot in the literature, that in the first experiment, people do it and they say, oh, there's no difference between males and females, so I'm just going to use males from now on, or I'm not going to analyze with sex as a factor. But just because you don't see a difference when you first are looking at something doesn't mean that there's not underlying mechanistic differences, or something comes along like age or disease or stress that might elicit a trait difference or a difference in the molecular mechanisms. The last point I want to make is that women's health is not going to be addressed by consistently comparing to men's health. Again, it's like comparing apples and oranges, but it's more than that. It's about not understanding that there's many types of apples and many types of oranges that can contribute to the heterogeneity. We already know that a number of these sort of female-specific experiences can influence health outcomes and disease risk. And I've studied almost all of these. I won't talk obviously about all of these today, but I've been really interested in how the effects of pregnancy influence brain health, both in the short term and the long term. I spent a lot of time looking at estrogens and how they influence brain health with an eye towards depression and aging and Alzheimer's disease. And I would say that the major points I sort of have learned is that building a model, it doesn't matter if you're looking at rats or mice or humans of disease, it's really important to incorporate who is getting the disorder and when. That's really important for translation. The other is to embrace that heterogeneity or complexity in your data and to rethink dogma. And now let's see if I convince you. So what do we learn when we're considering who is getting a disease and when? And I'm going to use the two examples of Alzheimer's disease and major depressive disorder. So what are the modifiable and non-modifiable risk factors for late onset sporadic Alzheimer's disease? We know that there's about 45% of Alzheimer's disease is contributed by lifestyle factors that are shown here. And the top three non-modifiable risk factors are advancing age, ApoE4 alleles, and female sex. And it turns out that sex plays a role in every single one of these factors. And I've been studying the interaction, and I have a number of papers out already looking at the interaction between ApoE4 alleles and female sex. I won't get to that today, but happy to answer questions about it. So I think it's always really interesting to study people that are at risk for Alzheimer's disease. So another population that's at risk for Alzheimer's disease is a population called amnestic mild cognitive impairment. And about 50% of these individuals would transition to Alzheimer's disease. However, more males are diagnosed, human males are diagnosed with amnestic MCI, and more females are diagnosed with Alzheimer's disease, as I've told you. So I'm like, well, what's going on here? Why is there this sort of paradox? And this is part of the sort of rethinking dogma. So it turns out that if you look at a number of cognitive domains, global cognition, executive function, verbal memory, females score higher than males do. And this is seen throughout life. If we're not using a sex stratified score at the end of the day to diagnose people, we might be missing females that have amnestic MCI. And that's exactly what Erin Sunderman found when she actually looked at sex as a factor. So when she used sex-specific norms to diagnose amnestic MCI and she looked at retrospective data, she could improve diagnostic accuracy by 20%. Improving diagnostic accuracy is huge, huge for the person, huge for your study. So she found 10% more females at risk and 10% fewer males at risk. And I think we also have to think about this in terms of biomarkers that people are trying to find to see what's predictive and what's not predictive. If we're not capturing the right population to begin with, it just makes our noise more messy. And so here's another example of why studying sex can be really helpful in studies. What about major depressive disorder? I put some common risk factors up there for major depressive disorder. Female sex is almost the number one. Chronic illness turns out to be the number one. Oh, but it turns out that females are more likely to suffer from chronic illness, so they're sort of mixed up within this variable. Family history, as well as factors that I'd call chronic stress. And in my lab, we've been looking at the interaction between female sex and chronic stress. But just thinking about the male to female ratio. So I've already told you that females are more than twice as likely to be diagnosed with depression, or I should have told you that. And this is the female to male ratio across the lifespan. So anything above 2 would be more than twice as likely. And what I think is really, really interesting is where it comes above 2 is a time of higher levels of reproductive hormones for females, suggesting that there's a unique physiology that results in these specific periods of susceptibility to depression across the lifespan. It also suggests that ovarian hormones might be contributing. Now, the graph I showed you suggests that it might be contributing to risk, right? There's a higher amount, so that means higher prevalence during reproductive years, ovarian hormones must be contributing. But if you look a little deeper, you'll see that the time of greatest risk to develop first-time depression is actually during the postpartum, when levels of ovarian hormones or placental hormones, in that case, are really, really low. And the same thing with perimenopause. That's the second time of greatest risk to develop first-time depression. And those levels are coming from sort of a high level to a low level. So it might be these fluctuations. One of the things that we've been studying is perinatal depression or postpartum depression in our case. And it turns out there's actually quite a bit of heterogeneity. And so this is the idea of embracing that heterogeneity in your data. And Alkista Skalkito, I used to talk about 2 different forms of postpartum depression. And she told me one day, you know, there's more than the 2 that you're talking about. And this comes from her clinical data. She's in Uppsala University, so very close to you. And I really appreciated her telling me this. So she sees healthy people that have normal sort of depression scores or lower depression scores, so no evidence of any depression. She sees people that have chronic depression throughout pregnancy and the postpartum period. She sees people that are only depressed during the pregnancy, only with an early postpartum onset, and only with a late postpartum onset. And many years ago, we started to create 2 animal models of postpartum depression, one with an early postpartum onset and one with a late postpartum onset. Now, I kind of want to make the point here that other people have looked at depression onset during pregnancy, and all of these models matter, right? It's not like one is better than the other because we know that the disease itself shows some heterogeneity, and so we do see differences even within our own model. So in terms of that early postpartum onset, we give a hormone stimulated pregnancy, then we withdraw the animals from this hormonal stimulated pregnancy and look at depressive-like endophenotypes. Or for the late postpartum onset, letting them have a normal pregnancy and then giving them really high levels of the stress hormone corticosterone just during that postpartum period. And we see so many interesting signatures of depressive-like endophenotypes. And what we found and others have found is that it's really important to embrace this heterogeneity because it turns out when you look, depression onset during pregnancy is more likely to be associated with a history of depression, whereas it's more likely to be associated with de novo or first-time depression in the postpartum. And as I said, as I show here, you can see different trajectories in terms of when the time of greatest risk is, the 3rd trimester versus early postpartum. And we also see differences in how antidepressants are efficacious towards the depression during these different time points. Those are really important to study. Now, I also get asked this question quite a bit. Don't men and women just show depression differently? You know, men don't want to talk about their feelings as much. And I really think that this is an important thing to consider. But let's look at how you diagnose depression. You have to have one of those 2 blue symptoms for at least 2 weeks, and then 5 out of the other 7 symptoms. This is for DSM-5. The European one might be a little different. And it turns out angst, I think, which is one of the best last names ever if you're in the business, looked at 78,000 interviews. And these are door-to-door interviews across 5 different countries in Europe. and as well as some community samples. So not just going to the doctor, this is like actually going into the community and asking. And it wasn't until there were 5 or more symptoms that that female to male ratio increased. So there's something about these symptoms of depression that are eliciting this 2-to-1 ratio. There actually tends to be 2 things that I want to point out. One is that it's a pretty heterogeneous disorder itself, right? You can have weight gain or weight loss. You can have insomnia or hypersomnia. You can have agitation or retardation. And so it does make it complex to model. There are also many other kinds of symptoms of depression that don't sort of make it into those classic lists. And it's been suggested there's 256 unique symptom combinations. Are there sex differences in symptoms of depression? It's actually pretty hard to find this data, which is also really interesting given that we've had these mandates for so long. But there is a suggestion, as I already told you, that females are more likely to show these atypical symptoms. They're more likely to show hypersomnia, hyperphagia, and more of those somatic complaints. So, what are the consequences with this sort of atypical label? And it started to get me thinking about how are clinicians measuring depression in the clinic? And they're looking at a variety of symptoms using these, there are 7 scales that are used. And it started getting me thinking, are those scales actually asking about those atypical symptoms? Because if they're not, maybe we're not capturing all the women that we should be capturing. So, and this is not a scientific look at this. I just quickly started looking at the scale. I really appreciated this study. This also comes from Leiden University, looking at the scales and showing that there was not that much overlap between the 7 scales in of itself. But if you look at the scales that are asking about these female-specific symptoms, you can see that there are not that many of them. The questions are not the questions are not being asked across all scales. So it starts to make me think: are we missing females because we're not asking about these sort of atypical symptoms? Okay, so that's diagnoses. What about some biomarkers? What do we lose when we're not considering sex as a meaningful variable? In terms of Alzheimer's disease, there's some significant neuropathology that you need to present with before being diagnosed with Alzheimer's disease, rather than just a dementia, and that's amyloid beta protein and neurofibrillary tangles. And it turns out that, and this is some data we did from a database, that females are in light blue, the males, so this is in humans, and the males are in darker blue there. And remember, I told you that if you possessed the ApoE4 alleles, you are at greater risk. And this is in cognitively normal people with amnestic MCI, as well as people that were diagnosed with Alzheimer's disease. And actually, diagnosis didn't play a large role here, but we saw that females with ApoE4 female ApoE4 carriers had higher levels of this phosphorylated tau and tau in their CSF levels, suggesting that they have disproportionately higher levels of one of these neuropathological markers. Now, other people have seen the same thing. That's Tim Holman, Rachel Bleckley, and all sorts of other databases. But what this also suggests to me is it's an example of a latent sex difference. There's no sex difference in the non-carriers, but genotype comes along, and that's when you see a sex difference. Now, you might have seen some of these headlines from a couple of years ago now, because an Alzheimer's drug was approved by the FDA. It's called lecanemab. There are some major side effects, but it did seem to slow some cognitive decline in certain people. Now, buried in this New England Journal of Medicine article is that, and it didn't focus on tau, it focused on amyloid beta, is that it actually was more effective in males than it was in females. It was more effective in non-carriers and heterozygotes of ApoE4. It was not effective in the homozygotes. And in terms of other factors, it wasn't effective, or it wasn't as effective in Black individuals as it was in white individuals. Now, I think this is really important and not something to be buried in a supplemental table. Right? If the people that are at greater risk are females and are homozygotes for ApoE4, we're not serving those individuals. I'm very happy for the white males that are non-ApoE4 carriers, but we need to find a medication that will work in other individuals that are more susceptible to Alzheimer's disease. The other thing I alluded to is that it had some severe side effects. So they came out with a list to see who's more susceptible to these side effects. But sex and gender wasn't considered. That's at Hung and all. And that's not great because if females are not served by this medication, but then they also have more severe side effects, again, we deserve to know that information. And just bringing it back to some data that we've collected ourselves, looking at sex and ApoE4 as a factor. This is in rats, this is in middle-aged rats, and I really care about the hippocampus. And I'm looking at here at putative neural stem cells with SOX2 and double cortin, which is an immature neuronal marker. And what we see in males, middle-aged males, is that neural stem cells or neuroprogenitor cells are decreased. The number of these new neurons are also decreased with the human knock-in of ApoE4. And this is exactly what you'd expect. This is the risk, the gene for risk for Alzheimer's disease, and we're seeing reductions in neuroplasticity, right? Reductions in the plasticity of the brain. But remember, females are the ones that are more risk, and they're actually even at greater risk with ApoE4 alleles. We see no difference in neural stem cells, and we actually see maybe the opposite to what you'd predict, that there are more of these new neurons in female ApoE4 carriers. And now, you might think, well, maybe this is just a bad model, which absolutely is a possible explanation. But another explanation is maybe it makes sense in the males because we've done so much research in males, and that the hippocampal neuroplasticity is disrupted in males. This makes sense, but maybe there's something else going on, or maybe there's another brain area that's triggered in females, or maybe those new neurons are not contributing in a meaningful way in females. And in fact, there is data from Jessica Caldwell's lab looking at female humans showing the exact same thing, that there's greater hippocampal integrity in the face of more pathology. Just turning to sex differences in depression biomarkers here, there are a number of signals that seem to be a little different. There's more leptin, more immune signaling differences, higher inflammation in females with major depressive disorder. But again, very hard to find these data. And I really hope I'm driving home the point that not using sex as a variable will hamper our understanding of what's going on with many of these disorders. A very nice study that came out showed that there were this is looking at transcriptional signatures in humans, postmortem tissue, showed that there was very little overlap in the signatures in a variety of brain areas, transcriptional signatures in a variety of brain areas. And even in the overlap here that's shown, the genes went in completely opposite directions. So that's why it's so important to keep examining sex as a factor. Because if we find a target, I don't know, let's say it's IL-6 or something, and we see that it's increased and we want to decrease it. Well, maybe that's only true in females, and you could be harming males, not helping males with depression. That's why it's so important to examine these factors. And I thought I'd give you another little tidbit if I have time to give you a little tidbit. This is looking at a cognitive bias task. And so um, in people with depression, they often show something called negative cognitive bias. And that is when you find an ambiguous situation or comment as negative, you're interpreting as negative. So Dr. Travis Hodges, who did a postdoc in my lab, he uses the example of somebody might say to him, "That's an interesting shirt you're wearing." And if you have a negative bias, you would think you'd be insulted that somebody thought that you had an awful, ugly looking shirt. But Travis is a very positive bias, so he's going to say, "Thank you very much. I like my shirt too." So how did we model this in animals? We had two contexts, one they got shocked in and one that they didn't get shocked in. And we gave them these two contexts every day for 16 days. Then we gave them a day of rest, and then we put them in an ambiguous context. So it had half the features of the shock context, half the features in the non-shock context. So they've obviously learned over 16 days to discriminate between the two contexts. They freeze more in the context they're going to get shocked in. They don't freeze in the context that they're not going to get shocked in. So what happens on the ambiguous day? And our interpretation was that if they showed an increase in freezing, that would suggest that they have more of a negative bias. And if they showed a decrease, that would suggest they have a neutral or possibly positive bias. And we looked at a number of biomarkers in this task. And I just want to show you some data we haven't published yet, showing that when we gave chronic unpredictable stress, which is one of the more common ways to model depression in animals, and this is over 14 days, we actually saw more of this negative bias. There's more freezing to the ambiguous context in those animals that underwent the kind of chronic unpredictable stress, and both females and males. So again, there's no sex difference here in terms of the amount of freezing. But what we found was very different neuroinflammatory signals, very different neuroplasticity signals, and very different functional connectivity. And I hope we're going to preprint this article very, very soon so you can see the full extent of what we did. But what just that's a little taster, that negative cognitive bias has different representation in females and males. We saw greater neuroinflammation, particularly in amygdala. We saw different neural networks. And this is very similar to what has been seen by Marianne Senley, who's at University of Pittsburgh in the US, and at Jen Sibyl, who's at CAMH. And also suggestive of a mechanistic difference, right? We didn't see a sex difference in negative cognitive bias, that freezing after chronic unpredictable stress, but we're seeing that the mechanisms underlying it are very different. Now, we also see, and tell me, please give me more examples of this, because I did this very quickly, but we also see sex differences in drug efficacy everywhere. 90% of women experience more side effects than men. And I think this is information that shouldn't be hidden, but is hidden. I think this has to do sometimes with the regulatory procedures that people have to follow, that drug companies have to follow. But I think this is exactly what we need and should mean when we're referring to things like personalized medicine or precision medicine. I'm not sure what term is used in Europe. And I'm hoping that the takeaways you got from my talk are that males cannot serve as a default for females, as there's just too many differences that we see. I didn't give you all of these differences, but we see them in my own lab in terms of learning, neural activity, functional connectivity, inflammation. And the second takeaway is that even when you see no evidence in your for sex difference in your first experiment, that doesn't guarantee there's not going to be no further sex differences in mechanism. And I want to close by just saying, despite the mandates that are out there, we're not seeing much movement in the field. So this was a survey of over 3,000 articles in 6 journals, like Journal of Neuroscience, Nature Neuroscience, Biological Psychiatry. We looked at 2009, 2019. There's 3,000 articles we looked at. There were many more studies that were including both males and females. About 70%, that's quite good. But very few that omitted what sex they use. That's really fantastic. But only 20% use what we called an optimal design. And all we meant by that, one of the barriers was really low barrier. One was that they had an equal sample size, or that they use sex throughout all of the experiments. Perhaps more alarming is that only 5% of the studies actually used sex as a discovery variable or a meaningful variable in their analyses. So if you're not analyzing to see if there is a possible difference, we're no further along in our knowledge. As well, there were 9 times more male-only studies than there were female-only studies. I really like this study by Neil Epperson. This is looking at neuroimaging studies published in Neuropsychopharmacology. So she found in this, but a little better, that 20% of the articles actually examined whether sex made a difference or not. But of those, 72% found a difference. That's a pretty large number. So if you look, you're probably going to find. You might ask me about power. Happy to talk about it. The last little thing that I'm happy to answer questions about is that let's not ignore female or women's health. We have female unique experiences that influence health across our lifespan. And again, I study many of these. A barrier I see in women's health is that we overgeneralize a lot. And I don't know if you've seen this show. I think it's on Prime. It's on Prime in North America. Reacher. It's kind of funny, but he's not funny. It's kind of an interesting show. He's a sort of a detective, and he keeps talking about like details matter and the investigation details matter. They also matter in animal studies. They also matter in clinical studies. When we say things like hormone therapy or estrogen or pregnancy, menopause, perinatal depression, even perinatal depression, we're ignoring that these are plural terms. There are many kinds of menopauses, many kinds of pregnancies. And when we're using these singular terms, the details matter. We're going to run into issues. We know that it's not just these female specific factors that matter, but many of the traits within these factors that also have a significant effect on health outcomes and disease risk. And what happens when we ignore women's health research? It results in delays in diagnosis, as I said at the beginning. It's a missed opportunity for earlier intervention, and we know the optimal time to intervene for any disease is the earlier the possible. It results in slower adoptions of precision medicine. It costs lives, and not just the lives of women, but also men. And I'm happy to tell you why I think that. It also costs the economy. And there was a World Economic Forum report that was released last year suggesting that we could save the world economy $1 trillion a year, if we just understood women's health more than we do now. Women's health research is underfunded. It's true in Canada. It's true in the US. I'm sure it's true in Europe. What can we do about it? We can get loud. That's why I do these kinds of talks, and not just for a fellow Estonian, but also because I care really deeply about it. So if I have moved you, or please spread the word. And I'm happy to talk about that more. We can invest in women's health research. So if you know any policymakers or people with lots of money, please tell them to invest in this. I think about the ALS Ice Bucket Challenge. They raised 115, I think it's 114, but it doesn't matter, million in 2014. That doubled the number of publications. That's just 5 years later, so it's going to be more even now. Doubled the number of researchers, tripled the amount of funding, and there were 10 times the number of clinical trials in 7 years. That's amazing progress. And the other thing is that I'd encourage you to join. I created something called the Women's Health Cluster. It's free to join. Whereas 32 countries, we're on a mission. It's to promote, expand, and catalyze women's health research. I want to connect like-minded people. There's a lot of free information we have. We have a seminar series, we have a podcast, we have a blog. Please contribute. We're really happy to have any suggestions or contributions from people as well. And just to conclude, I think and I hope I've given you some ammunition for this too, that understanding basic neurobiology and disease mechanisms in males, females, and intersex individuals will lead us to better individualized treatments for disease. Obviously, intersectionality matters. And, I know what's happening in the US right now. I hope it doesn't infiltrate into other countries. But, we ignore these things and we ignore these differences at our own peril. It will lead to worse outcomes. And with that, I just like to acknowledge the people that actually do the work in my lab and the funding agencies, past and present, that have given me money to do some of this work. And the last thing I'll say is Organization for the Study of Sex Differences. I was a past president. I'm the past president. And it's in Albuquerque. Next year is going to be in Mexico. Not Mexico. Next year is going to be in Hawaii. That might be a more fun place to go to. I shouldn't say. I'm sure New Mexico is going to be great, but please consider it. And I'm hoping the year after we'll be in Europe again. And thank you very much. I'll stop sharing. So hopefully there's some time to ask questions.